Hello everybody, my name is Emily Fox and today we will be discussing the processes of hemostasis and thrombosis. This material can be found in Harper's Illustrated Biochemistry, the 31st edition in Chapter 55. Alright, we have 5 learning objectives for today. By the end of the lecture, we will be able to describe the 3 phases of hemostasis and explain how they contribute to the cessation of bleeding. Differentiate between types of thrombi and their clinical significance. Explain platelet activation and aggregation, including the role of thrombin and endothelial regulation. Compare the intrinsic and extrinsic coagulation pathways and identify the factor that links them. And lastly, we will be able to recognize therapeutic strategies and genetic disorders that affect coagulation and fibrinolysis. All right, to start us off, why is hemostasis and thrombosis important to learn and understand? One of the major causes of serious medical emergencies is due to hemorrhagic and thrombotic occurrences. It is important to have a clear understanding of these terms and their relative mode of action in the body to fully manage related conditions. Some common conditions may include, but are not limited to, strokes, myocardial infarctions, also known as heart attacks, and pulmonary embolisms. So, what is the difference between hemostasis and thrombosis? Hemostasis is the cessation of bleeding from a cut or a severed vessel and occurs as a natural response to a vascular injury. While on the other side, we have thrombosis, which occurs when the endothelial lining of blood vessels is damaged or removed. This is an abnormal response to endothelial damage. These 2 processes share 3 common phases. In phase 1, we see the formation of a loose and temporary platelet aggregate at the site of injury, while in phase 2, we see the formation of a fibrin mesh that binds the platelet aggregate, forming a more stable hemostatic plug or thrombus. In the last common phase, or the 3rd phase, there is a partial or complete dissolution of the hemostatic plug or thrombus by plasmin. There are 3 types of thrombi, or blood clots, in which all 3 contain fibrin in various proportions. A white thrombus is composed of platelets and fibrin and is relatively poor in erythrocytes. They form at the site of injury or abdominal vessel wall, particularly in areas where blood flow is rapid, such as arteries. Red thrombi consist primarily of red blood cells and fibrin. They resemble the clot formed in a test tube and may form in vivo in areas of slowed blood flow or stasis, such as veins, with or without injury. Lastly, we have fibrin deposits, which appear in very small blood vessels or capillaries. All right, so let's now talk about platelet activation and aggregation. Platelets normally circulate in an unstimulated disc shape. During hemostasis or thrombosis, platelets become activated and help form hemostatic plugs or thrombi. This process occurs in 3 major steps, the first being adhesion to exposed collagen in the blood vessels. At this point, platelets bound to collagen change shape and spread out on the subendothelium. In the second step, we see a release, or exocytosis, of their contents of their storage granules. Some of the molecules released in this stage will amplify the responses to vessel wall injury. Polyphosphate, or one of the molecules released from the dense granules, accelerates factor V and factor XI activation by thrombin. In the last major step, we see aggregation. The aggregating agents, or thrombin, collagen, ADP, and others, such as the platelet activating factor, use an inside-out signaling pathway to modify the platelet surface. This happens so the receptor has a higher affinity for fibrinogen, or the von Willebrand factor. Something else to note is that thrombin, which we will later see in the coagulation cascade, is the most potent activator of platelets and initiates activation by interacting with its receptors on the plasma membrane. Activated platelets, besides those forming a platelet aggregate, accelerate the activation of the coagulation factor X and prothrombin by exposing the anionic phosphatidylserine on their membrane surface. Along the walls of blood vessels, we can find endothelial cells. These cells make important contributions to the overall regulation of hemostasis and thrombosis. They synthesize the PGI2, a potent inhibitor of platelet aggregation that acts by stimulating the activity of adenylyl cyclase in the surface membrane of platelets via its G protein coupled receptor. Endothelial cells also possess an ADPase, which hydrolyzes ADP, therefore opposing its aggregating effects on platelets. As an aid in dissolving thrombi, endothelial cells express proteoglycans such as heparan sulfate, an anticoagulant, which releases plasminogen activators. Which something really interesting about endothelial cells is that if damage were to occur to them, they are able to regenerate within hours. Antiplatelet drugs are medications used to prevent platelets from sticking together and forming blood clots. These drugs are very common, with the most common being aspirin or acetylsalic acid. Platelets are very, very sensitive to aspirin, and as little as 30 milligrams, which is about the weight of a grain of rice, of aspirin a day effectively eliminates the synthesis of TXA2 by platelets. Indications for aspirin use include, but are not limited to, management of coronary syndromes such as angina or myocardial infarctions, acute stroke symptoms such as transient ischemic attacks or acute strokes, severe carotid artery stenosis, and primary prevention of these and other thrombotic diseases. All right, there are two pathways that lead to fibrin clot formation, the extrinsic and intrinsic pathways. These pathways are not independent and both lead to the proteolytic conversion of prothrombin to thrombin. Thrombin is important because it catalyzes cleavage of fibrinogen to initiate fibrin clot formation. Both of these pathways are complex and involve many proteins, but the link between the two is factor 10, and this is very important to remember later on. All right, let's start with the extrinsic pathway. The extrinsic pathway is rapid and occurs when there is external trauma. When a tissue injury occurs, a tissue factor or a protein is exposed and the process of fibrin clot formation is initiated. The tissue factor is where the extrinsic pathway begins. This tissue factor activates factor 7, a protein in the blood, and then it becomes factor 7A. From here, we have the tissue factor, factor 7A, and now we add calcium, which forms a team called the extrinsic tinase complex. This team is responsible for activating factor 10, turning it into factor 10A. Factor 10A is the key enzyme that aids in building blood clots. The body naturally keeps this pathway in check through the tissue factor pathway inhibitor, which is basically a brake for the blood. Its role is to block factor 10A, stopping it from making too many thrombi. All right, now let's talk about the intrinsic pathway. It is slower and way more complex, taking longer to activate due to the need for additional factors. It is simply another route the body uses to make factor 10A. This pathway is activated by contact with negatively charged surfaces, such as damaged endothelial cells or exposed DNA. This exposure activates factor 12, converting it to factor 12A. At this point, factor 12A then activates prekallikrein to become kallikrein, which loops back around to further amplify the activation of factor 12. The cascade continues as factor 12A activates factor 11 to become factor 11A, to then activate factor 9 to factor 9A, with calcium as a necessary cofactor. It gets a little confusing, but it's a very necessary process if the extrinsic pathway is not working. On the surface of the platelets, we see factor 9A combining with factor 8A, which is an essential cofactor rather than an enzyme, and calcium to form the intrinsic tinase complex. Much like the extrinsic tinase complex, this complex is responsible for activating factor 10 to factor 10A. Something unique to point out about the intrinsic pathway is that it, although contributes to clot formation, it is not always essential. Meaning that if an individual lacks certain intrinsic factors, they will not necessarily exhibit abnormal bleeding. Instead, this pathway plays a bigger role in amplifying clot formation and potentially aiding in clot breakdown. So how do these two pathways connect? Like I mentioned earlier, the intrinsic and extrinsic pathways connect through factor 10, as seen in the picture shown. Once factor 10A is generated through either pathway, it becomes central to a common pathway. Factor 10A, along with factor 5A and calcium, form the prothrombinase complex. This complex catalyzes the conversion of prothrombin into thrombin. Prothrombin is a single-chain glycoprotein that is synthesized in the liver. Once it has bonded to the prothrombinase complex, to which it becomes cleaved into active 3-chain thrombin molecule. Factor V, a component of this complex, is found in both plasma and platelets and is synthesized in the liver, spleen, and kidneys. It becomes activated once it meets traces of thrombin, ultimately creating a feedback loop that further accelerates clot formation. With all of that said, thrombin plays a huge role in stabilizing the clot and activating the downstream components of the coagulation cascade. As we see the final stages of the coagulation cascade, we see thrombin play an essential role in converting fibrinogen to fibrin. Fibrinogen, a soluble plasma glycoprotein, becomes insoluble fibrin, essentially forming a clot. Fibrinogen is highly abundant in plasma and contains a dimer made of 3 distinct polypeptide chains that are covalently linked by 29 disulfide bonds. All 3 chains are synthesized in the liver, and the 3 genes encoding these proteins are located on the same chromosome, allowing for coordinated regulation of their expression in humans. There is an abundance of aspartate and glutamate residues within these chains that result in a high negative charge. The negative charge contributes to the solubility of fibrinogen in plasma and plays an important role in preventing aggregation by causing electrostatic repulsion between fibrinogen molecules. In addition to converting fibrinogen to fibrin, we also see thrombin activating factor VIII to factor VIIA. This activated factor is a highly specific transglutaminase that covalently cross-links chains of fibrin molecules by forming peptide bonds between amide groups of glutamine and the amino groups of lysine residues. The cross-linking that occurs yields a more stable fibrin clot with an increased resistance to proteolysis. This fibrin mesh that forms serves to stabilize the hemostatic plug or thrombus. Alright, let's talk about the regulation of thrombin. One active thrombin is formed during hemostasis or thrombosis, but its concentration must be carefully controlled to prevent further fibrin formation or platelet deactivation. We see this process happen in 2 different ways, the first of which is feedback mechanisms. Here we see a delicate balance of activation and inhibition, and the clotting cascade provides amplification. The second way is circulating inhibitors such as antithrombin. There are 4 naturally occurring thrombin inhibitors that exit within normal plasma. This consists of antithrombin, which is the most important as it takes up 75% of our natural anticoagulants. Next, we see alpha-2 macroglobulin, which takes up about 20%. In the last 5%, we see our minor inhibitors such as heparin cofactor 2 and alpha-1 antitrypsin. But let's look into some of the anticoagulants that we give. So we have low molecular weight heparins, also seen as LMWHs, are derived from enzymatic or chemical cleavage of unfractionated heparin with a higher clinical use. There's a nice thing about these drugs, and it's actually that they're able to be administered subcutaneously at home. They do not require frequent monitoring and have a greater bioavailability than unfractionated heparin. Next, let's look at the coumarin drugs such as warfarin. These drugs inhibit the reduction of the quinone derivatives of vitamin K to the active hydroquinone forms. It is important to note that this drug is reversed with vitamin K, but it can take up to 12 to 24 hours to do so, but can be done faster by infusing normal coagulation factors. Lastly, let's look at thrombin, which is a key enzyme in coagulation. Thrombin is a part of a natural anticoagulant pathway that slows down thrombin generation, therefore preventing excessive clot formation. This is done when thrombin binds with thrombomodulin, which creates a complex that activates protein C, or APC. When APC has protein S as a cofactor, it inactivates factor VIA and factor VIIA, thus regulating clot formation. When there is a deficiency in protein C or S, this natural anticoagulant pathway is disturbed and it leads to a greater risk of venous thrombosis, such as DVTs or PEs. In conditions as such, we tend to use heparin and warfarin used as treatment, along with prophylactic measures in patients with strokes, AFib, and other heart rhythm anomalies. All right, let's talk about some genetic bleeding disorders. There are many genetic bleeding disorders that are inherited deficiencies of the clotting system that result in abnormal bleeding in humans. Most commonly, we see a deficiency in factor VIII, which causes hemophilia A. This is an X chromosome linked disease, which I find quite interesting. We can also see a deficiency in factor IX, which causes hemophilia B. This is also an X chromosome linked disease and has been recently identified as the form of hemophilia that played a major role in the history of the royal families in Europe. Another fun fact: when looking at the most common hereditary bleeding disorder, we see von Willebrand disease. This is a result of a deficiency or defect in the von Willebrand factor. This factor is a large multimeric glycoprotein that is secreted by the endothelial cells and platelets into the plasma, stabilizing factor VIII. It also plays a role in promoting platelet adhesion at the sites of vessel wall injury. When we talk about therapeutic fibrinolytics, think medications used to dissolve blood clots. The main therapeutic fibrinolytic used is TPA, or tissue plasminogen activator. It is marked as alteplase and is produced by recombinant DNA methods. This medication is therapeutically used and widely indicated for myocardial infarctions, strokes, pulmonary embolisms, and many more. There are several disorders, including sepsis and cancer, that naturally increase the concentration of plasminogen activators within the body. There are also circumstances such as liver cirrhosis, in which plasminogen activators are naturally impaired in the body. When treating processes as such, it is important to know how they affect these factors in order to proceed with adequate treatment. Fibrinolysis is the coagulation system that is normally in a state of dynamic equilibrium in which fibrin clots are continuously being formed and dissolved. Plasmin, or the serine protease that is responsible for degrading fibrin and fibrinogen, circulates in the form of plasminogen. There are various types of plasminogen activators, and they are found in most body tissues. They all cleave the same ARG-VAL bond in plasminogen to produce disulfide bridge-linked two-chain serine protease, plasmin. When it comes to regulation mechanisms, we see that plasmin targets fibrin specifically, which in turn limits off-target degradation. We also see TAFI-A, or activated thrombin activable fibrinolysis inhibitor, remove terminal lysines from the fibrin, which allows it to inhibit fibrinolysis during clot formation. There are numerous lab tests that are available to measure the phases of hemostasis in different capacities. Here is a list of a couple. Some really important ones to look at are platelet count, bleeding time, closure time, platelet aggregation. We tend to look at prothrombin time or thrombin time a lot, and also the measurement of fibrin degradation products. So, overall, what did we learn today? We learned that hemostasis and thrombosis are very complex processes that involve platelets, coagulation factors, and vascular components. We learned about natural and external coagulation mechanisms such as heparin and antithrombin. We also learned about the coagulation cascade and the two processes within this process. One of the most important takeaways is knowing why this information is important to know and understand. Hopefully, after this lecture or PowerPoint, you can do just so. All right, let's get into some questions to test our knowledge on what we learned today. Question 1, we have, which of the following best describes the first phase of hemostasis? A, partial or complete dissolution of the hemostatic plug or thrombus by plasmin. B, formation of a loose and temporary platelet aggregate at the site of injury. C, formation of a fibrin mesh that stabilizes the clot. Or D, conversion of fibrinogen to fibrin by thrombin. The correct answer here is B, formation of a loose and temporary platelet aggregate at the site of injury. This is the first of 3 common phases between hemostasis and thrombus that we learned earlier. If you look, the second phase is actually answer number C, formation of a fibrin mesh that stabilizes the clot. And the third phase is A, the partial or complete dissolution of the hemostatic plug or thrombus by plasmin. All right, question 2. Which of the following best describes a red thrombus? A, consists primarily of red cells and fibrin. B, appear in very small blood vessels or capillaries. C, made exclusively of von Willebrand factor. Or D, primary platelets and fibrin, poor in red blood cells. The correct answer here is A, consists mainly of red blood cells and fibrin. If we look at B, appear in very small blood vessels or capillaries, we can see that this describes fibrin deposits. And then if we look at D, primary platelets and fibrin, but poor in blood cells, we see that this is describing a white thrombus. All right, question number 3. Which factor links the extrinsic and intrinsic pathways of coagulation? A, factor 5, B, factor 8, C, factor 10, or D, factor 4? The correct answer here is C, factor 10. The intrinsic and extrinsic pathways work independently up until they reach the common pathway, which begins with factor 10. These pathways are vital in order for our bodies to be able to form a stable blood clot. A deficiency in this enzyme can lead to excessive bleeding, making it incredibly significant. All right, question number 4. Which of the following mechanisms helps regulate thrombin activity to prevent excessive clot formation? So basically, what does our body do to help prevent too many clots from forming? A, platelet activation reduces thrombin generation. B, factor 8A cross-links fibrin to stabilize the clot. C, thrombin directly converts plasminogen to plasmin. Or D, antithrombin inhibits thrombin activity in circulation. The correct answer here is D, antithrombin inhibits thrombin activity in circulation. Antithrombin is responsible for regulating clot formation by neutralizing thrombin, therefore reducing the risk of thrombosis and maintaining hemostatic balance. Let's briefly look over why the other options are incorrect. So for the first choice, we see platelet activation reduces thrombin generation. Actually, platelet activation promotes thrombin generation by providing a surface for coagulation complexes to assemble. For B, we see factor 8A cross-links fibrin to stabilize the clot, which is exactly what it says. It stabilizes or strengthens the clot, but does not necessarily regulate thrombin activity. Lastly, we see C, thrombin directly converts plasminogen to plasmin. Thrombin does not directly activate plasminogen, as that is the role of TPA. All right, our last question, question number 5. Let's look at a patient, a little clinical situation here. A patient presents with easy bruising and prolonged bleeding. Labs show a deficiency in factor 8. What is the most likely diagnosis? A, hemophilia A, B, hemophilia B, C, von Willebrand disease, or D, protein C deficiency. All right, the correct answer here is A. So let's get into why A is correct. When we see a patient presenting with easy bruising and prolonged bleeding, we are automatically going to think about a genetic bleeding disorder. As we mentioned earlier, these disorders present with a deficiency of the clotting system that can present as a patient who bleeds very easily or too easily. To differentiate which genetic bleeding disorder, we need to focus on which deficiency they have. In this case, they have a deficiency in factor 8, which if we recall what we learned, we know that this deficiency is found in hemophilia A, which is the answer to this question. Lastly, here is my reference. If you guys have any questions or you want to look into greater detail about some of the things that we talked about today, feel free to read in our book. And thank you all for your time today.